Well, the LIGHTHOUSE study was a phase 3 prospective study from Blue Earth Diagnostics evaluating a special PSMA tracer, fluorinated PSMA radiohybrid PSMA 7.3. That was the name of the tracer. And it evaluated in patients, in primary prostate cancer patients who were scheduled for radical prostatectomy. And they had to have unfavorable intermediate risk features at least, or high risk prostate cancer features. And the primary endpoint was, it was a split endpoint between sensitivity and specificity of this fluorinated radiohybrid PSMA 7.3 tracer for detection of lymph node metastasis in the patient. And histology served, of course, as gold standard. Also, I have to say, with other PSMA tracers, and that are reported in literature already, this tracer basically, performs as good as the other tracers, but increases, of course, significantly the detection when you look at conventional imaging. This was not really looked at in the study, but when you look at the histopathology, the lymph nodes that you detect, the specificity of PSMA-PET, and especially this tracer, was also very high, 96% specificity for detection of lymph nodes. For the sensitivity, the maturity read was 24%, which I think also is not bad, because mostly these are small lymph node metastases. And so it confirms a little bit also the pro-PSMA study by Michael Hoffman that also showed a very good high specificity. However, in this study by Michael Hoffman, only a little bit over 80% received radical prostatectomy and had histopathology as gold standard, also included roughly 300 patients. And in our study, in the LIGHTHOUSE study, we actually have the full data set for 296 patients who, after PSMA-PET underwent radical prostatectomy and extended pelvic lymph node dissection. So it's, I think, the quality of the data here is really robust. And it again confirms the value of PSMA-PET for detection of lymph node metastasis in primary prostate cancer. And when we talk more specifically about this specific tracer, I think it's very interesting. It's a very interesting tracer, as it can be labeled with different radiometals. So it can be, in the study was fluorinated PSMA tracer, which is really nice, gives you nice images compared to gallium-based tracers because of the positron range giving you the signal for the PET imaging. But also it can be labeled with, for example, radiometals that are used then for therapy. So you're having the same specificity of the molecule detecting PSMA, but you, on the other hand, can label it with different radiometals. That is one point. The second point, I think, which is also very interesting, specifically for this tracer, that it shows low renal excretion. So you have not a lot of bladder background or background in the pelvis, because there is no activity. And with several other tracers, you do have renal excretion, and this is, of course, more important for when you image biochemical recurrent prostate cancer patients, where you might want to detect a local recurrence or rule it out. And this was also looked at in another study, the Spotlight study, which is not presented now here, but generally these are two features that are very interesting for this tracer. Furthermore, fluorinating is also enables to produce the tracer and bring it to other institutions for imaging. And with gallium, you have to produce it basically on site because of the low half-life. Right now we do have advanced imaging. We detect disease earlier. We detect metastatic spread earlier. Also in this cohort, roughly 20%, depending on the reader. There were 3 blinded independent readers in the study. Also a good sign of a good study, I would say. But in 20% metastatic disease was detected, which would not have shown up on conventional imaging, most likely. So what do we do now with these new imaging showing us metastatic disease? I think for now we have to stick to basically the knowledge we have obtained from old conventional imaging and should not modify our treatment on this novel advanced imaging results too much. But we should also take it into consideration. We don't know if we're now treating better, but I think we can target the disease better. If this translates then in better outcomes, I would assume so. However, this data will not be available for the next 5-10 years or so. But it will impact definitely prostate cancer treatment.